All right, I think we should get started now. Um, It is really an honor to introduce Dr. Shibi Likhite, who is a research scientist at Nationwide Children's Hospital, which is in Ohio, in Columbus. He has over 12 years of experience in the field of gene and cell therapy, intrathecal gene therapy programs, which means putting it into the spinal fluid. Um, his work has been in developing gene therapy programs for treatment for multiple neurologic diseases, including the recently approved phase 2A clinical trial for spinal muscular atrophy, with spinal muscular atrophy with respiratory disease, distress type 1, SMARD1. He has also been instrumental in developing the gene therapy for different cancers, in particular for this audience, vestibular schwannomas. Thank you very much for joining us today.
Good afternoon, everyone. I hope I'm audible. First of all, I would like to thank the Children's Tumor Foundation for giving me this opportunity to be here today. Special thanks to Kate, who has been very patient with me all throughout this time. And it's just been amazing. This is the first time I've been to a CTF event, and it's literally from the beginning of the welcome speech till now. It is so much electrifying energy in the room. It is so inspirational, and there is so much love that I'm very glad to be part of this family. So today I hope I'll be able to take you through the journey of gene therapy landscape. These are my disclosures. So I have divided today's topics in four main categories. First, we will be spending majority of the time on the overview of gene therapy, talking about the basics of gene therapy, the challenges, and how the field is overcoming them. Then I want to introduce you to some of the gene therapy developments we have been able to do at Nationwide Children's Hospital. And then towards the end of the talk, I would like to touch on our small efforts on developing gene therapy for NF2 and in vitro disease modeling to study NF2 in the lab. So let me start with the first question that was on the app. And I'm sorry, I didn't know how to answer it on the app itself, but it was, what is gene therapy? So in its simplest terms, it's transfer of genetic material to treat or prevent diseases. Now, it can be applied in three different categories. The first one is gene replacement, where you are adding a healthy copy of an otherwise non-functional gene or a gene with reduced function. So in case of cancer or context of cancer, that could be a tumor suppressor that's a faulty. The second way to implement gene therapy is with gene silencing, which involves reducing the expression or abundance of an aberrant toxic RNA or DNA or protein, sorry. So in this case, it would be an aberrant oncogene. And the third way the gene therapy can be implicated is in case of gene correction. So the gene correction involves in vivo correction of mutation on the DNA level, as well as ex vivo correction of the mutation on the cells that have been isolated from the patient and put back. And this can be achieved with the help of technology that is very well known as the CRISPR. There is another type of gene correction that can also be done on the level of mRNA, and that is using either exon skipping or splicing modulations. So today we will mainly be focusing on the gene replacement part of gene therapy. Now, there are different types with which the gene therapy can be delivered. The main two are the using viral systems or the non-viral systems. And in today's talk, we will be focusing on the viral systems itself. So let's first go through why viral gene therapy as compared to the other non-viral treatments. For the most obvious reason, it is one of the most efficient and most long-lasting therapy because viruses have naturally evolved to infect human cells. So we can utilize that potential to deliver the gene therapy. At the same time, it also makes them with the highest risk. The viral gene therapy is also an option when no other treatment options or there is any room for any substantial improvement with any other treatments, especially in case of severe debilitating disorders where the benefits of viral gene therapy would outweigh the risks. The viral gene therapy is also helpful when the location or even though there is a wide distribution of the affected cells, which is otherwise impossible for other treatments to even reach to deliver the cargo that is needed. And as I mentioned earlier, viral therapy has the best potential for a long lasting effect after a single treatment. So when we are talking about viral gene therapy, there is a difference between what the natural viruses are and what the gene therapy viruses or gene therapy vectors are. So in case of a gene therapy vector, the viral genes are replaced with the therapeutic gene that we would like to deliver. Now, this therapeutic gene would be placed under the regulatory elements called or control elements. So in this scenario, the viral particle surface or the capsid actually acts as a carrier just for the delivery, where the cargo is our therapeutic gene. And so when we refer to these, we refer to them as either recombinant viruses or the viral vectors. So let's go through what are the main challenges for gene therapy. The first one being pathogenicity. What it means is the ability of a virus being used for the vector to actually cause the disease in humans. The second one being immunogenicity, that is the immune reaction once the vector is being administered or even once the transgene is being expressed. Third one being oncogenic potential, that is after the delivery of the virus, whether that vector can cause mutagenesis due to aberrant insertion into the genome. Third one being tropism. What it means is what type of cells the virus can target. We would like the gene therapy vector to target the cells that are relevant to the disease and not the cells that are not needed. The additional component to that also is specificity. What it means is expressing the transgene or our cargo again in the disease relevant cells. So that means how to avoid the off-target effects of the gene therapy. The next one comes on depends upon different viruses that you are using, and that is the packaging capacity, which is the size of transgene or size of cargo that can be packaged into the vector system. The next one, which is which also has more importance, is the pre-existing immunity. What that means is whether the existing immune system or the existing immune response in the patient can neutralize the vector even before it gets a chance to actually be functionally active. One of the main thing that is, or one of the main challenge of the gene therapy, especially the viral gene therapy that needs to be noted, is it is not reversible. What it means is once the gene therapy is delivered, it is forever. You cannot stop taking it or you cannot take it back, which causes a extra problem, which is actually behind this with respect to redosing. So for the patients that have already received the first type of gene therapy, because the immune system does get activated after that, the redosing of the patients with either the same or similar vector could be a problem. So there are a number of gene therapy vectors that are currently in clinical trials, ranging from retroviruses, lentiviruses, which are the integrating viruses, and AAVs, HSVs, and adenoviruses, which are episomal. That means they do not integrate into the host genome. So for today's talk, we will mainly be focusing on the AAVs. So what's an AAV? AAV is a adeno-associated virus, which is different than the adenovirus. So in this picture, this red dot is an adenoviral virus, while these smaller green dots are the adeno-associated viruses. So this is the electron micrograph picture of these viruses together. Now, what is the difference between the two? The adenovirus is a pathogenic virus. So it could actually have detrimental effects if it is actually utilized not in a correct way. However, the adeno-associated viruses are the non-pathogenic viruses that travel with the adenovirus. Now, as you would see in the upcoming slides, these viruses have remarkable safety. They can provide long-term expression, and there are more than 100 clinical trials in which the AAVs are being utilized. So what makes AAV a suitable gene therapy vector? As I mentioned, these are small replication defective viruses. They can infect both dividing as well as non-dividing cells. Mainly, they have very low frequency of host genome integration. They lack pathogenicity. They have very low immunogenic potential and no cytotoxic response. And one of the major advantage of AAV is that they are also suitable for large scale vector production, which I'm sure will actually be clear just in a few slides. So till now, the first three challenges that we encountered earlier can be rectified with the AAV. Now, again, when we are talking about AAV that are used in the gene therapy, we are not talking about the virus. We are again talking about AAV vectors. That means the viral genome is again replaced by the therapeutic gene under a control element. And again, we are using the AAV capsid as a carrier to deliver the cargo. Now, let's see how this AAV actually works. The vector that actually has the transgene inside, once delivered to the humans or even to the animal models, enters the cell by binding to the cytoplasm. The vector then enters the nucleus where the capsid is degraded and the DNA is released. This DNA can then transcribe into the RNA. This RNA is actually taken back to the out of the nucleus where it can form our therapeutic protein, which would then exert its beneficial effects. Now, as I mentioned, we were already able to rectify first three challenges using AAV: the pathogenicity, the immunogenicity, the oncogenic potential. What the next challenge being the tropism or the cell type that can be targeted. The AAVs do come in different flavors, or as they are scientifically known as serotypes, that actually allow targeting of different tissues and cell types. And that targeting or those differences are mainly based on the variations in the surface proteins of the AAV capsid. Now, with different AAVs and different AAV serotypes that are naturally occurring in nature itself, you can utilize a different AAV serotype based on the organ that or the tissue that you are trying to target, also the different cell type that you are trying to target. Again, an added level of specificity can be provided by utilizing a control element that is specific to the cell type that you actually want your therapeutic cargo to be expressed. So that way you actually have the tropism and the specificity going hand in hand to reduce any off-target effects. There is also another way of generating newer AAVs with a technique called AAV capsid evolution. What this technique allows researchers is take the naturally occurring AAVs and move them into even strengthening the tropism or detargeting a particular tissue if it is not needed. In majority cases, it is the liver, because for most of the viruses, the liver toxicity is one of the main components that is part of the immune response and the adverse effects. So with all these advantages, of course, AAV could be one of the preferential vector for the gene therapy, but it also comes with its own limitations. And one of the main limitations for the AAV gene therapy is its packaging size. As the AAV capsid can only hold the genetic element that is 4.8 kilobases long, which is 4,800 or 4,800 base pairs in case of a single-stranded AAV, or there is another way in which this vector works, which is a double-stranded AAV, where the first strand is complementary to the second strand, so it can actually it can bind to itself once it is outside the capsid and in the cell, but that also makes it the packaging capacity even half. So now you actually have a packaging capacity of only 4.8 kb. So what that means is any transgene that is bigger than 4.8 kb will not be able to be packaged in a single AAV, for example, NF1. The other limitation or the other challenge when it comes to the AAV gene therapy also involves the potential immune responses. As I mentioned, if the patients, because these AAVs do occur naturally and you can be pre-exposed to these naturally occurring AAVs, patients and all of us actually can have the presence of antibodies that are called as either binding antibodies or neutralizing antibodies that can actually neutralize your gene therapy vector once it is administered. Once the vector is administered, because it's a virus, it can still have the immune response, and the severity of that immune response is then dependent on the dose of the virus. And so even after the delivery of the vector, you can still potentially have the adaptive immune response that can make the gene therapy a little bit more problematic for redosing, as I mentioned earlier. Now, there are certain ways in which the field has been trying to rectify these by utilization of steroid administration or immunosuppression, neutralizing antibody or antibody exclusion criteria. So these are actually implemented in clinical trials to pre-screen the patients to make sure that they do not have the neutralizing or the binding antibodies at a certain titer. And also the newer techniques coming as plasmapheresis, where you can reduce the amount of circulating antibodies within a patient with the help of plasmapheresis technique. So with the given advantages and some of the limitations of AAV, it is still one of the best available vector for gene therapy. And what has been really seen is since its discovery in 1965 and the first clinical efficacy shown in 2008, there is an explosion of clinical trials using AAV in multiple different diseases. With the first approval of Glybera in 2012 for EMA, the next approval of Luxturna in 2017, and then the third approval of Zolgensma in 2019. So let's just quickly look at the AAV landscape as far as the clinical trials are concerned. As of 2022, there are 136 unique clinical trials involving 134 AAV drug products and treating 55 different diseases. Most of these trials are in the phase 1 or phase 1/2 phases, which is mainly to determine the safety of the AAV treatment. And again, as you can see, most of them are also dealing with the gene replacement strategies. When it comes to the AAV serotypes, certain serotypes are more commonly used within the clinical trials. As you can see here, mainly AAV2, AAV8, AAV9, and also certain control elements are more used to drive these therapeutic genes, mainly CAG, CBA, CMV, and these are the control elements that actually provide ubiquitous expression. So they act and ubiquitous high expression of the therapeutic molecule. If you consider the immunological strategies that are implemented in the current clinical trials, most of them include the steroid application and the NAB exclusion, which is what we just discussed, as the potential use of steroids during the study period and exclusion of the patients that have pre-existing anti-AAV capsid antibodies in at least for now. So if you see the current AAV landscape within past 20 years, most of the AAV clinical trials are in the phase of phase 1 or 1/2. But in the next 5 years, over 50 trials would reach the next phases of phase 3 and hopefully go into the market. So the take-home message so far is, of course, gene therapy comes with unique set of challenges that do need to be considered for the clinical trial design. Most of the programs, as we saw earlier, are in the early stages, so there is still a lot of learning to occur. As you can see, there are many AAVs available, and all of them seems to be working in animal models, but there are much fewer AAVs that can actually make the transition to the clinic. And that is one of the main component or main fact to be kept in mind when we are designing the studies that go from preclinical to the successful transition, that there are significant targeting differences between animal models, different tissues, and when it actually comes to the patients. Now, even though there have been these many AAV clinical trials, not all the clinical trials have been successful. And let's actually look at some of the potential reasons for the clinical trials that haven't been as successful in clinic as they have been in the animal models. One of the main things that would be very obvious is the drug just does not work. So in that case, you actually have to start again with a different drug or different drug product. It is also possible that the drug only works in a subpopulation of patients and not all the patients. It is also possible that drug would work if the timing of the drug administration and even the dosing of the drug administration is adequate. It is also possible and actually had seen that some of the outcome measures that are set into the clinical trial design don't necessarily reflect the efficiency of the treatment. This is mainly occurring or would occur with the rare diseases where the disease course is variable between different patients, as well as the disease. So the disease outcome measures are not set in stone. So because it actually changes so much, and your timelines for the actual outcome measure need to be determined before the start of the clinical study, you may miss them, which is where having a natural history cohort or at least a natural history of the patients for the disease that you are providing the gene therapy is extremely important. It's the same similar way as the outcome measures may reflect the efficiency, but the timing of that measurement is wrong, which is again talking about how you need to have, or it is beneficial to have a natural history for that particular disease. And finally, again, it comes to the selection of patient population, as in certain patients or certain mutations might not be responsive to the therapy that you actually are targeting for. Now, there is also a realization that is important when it comes to these gene therapies, and that is whether you're looking for stabilization of the symptoms or the complete reversal of the disease. In both cases, it is very disease mechanism dependent. It also depends on whether the disease that is in question requires rescue of cell survival versus rescue of cellular function. For example, in case of neurodegenerative diseases, if you are trying to target motor neurons, if the treatment is actually delivered when most of the motor neurons are dead, you do not have a cell to actually improve the cellular function because you have already removed your target cell type. However, if you actually are talking about a treatment for a disease like Rett syndrome, where there is not a neuronal death, but the neuronal function is affected, introducing a cell gene therapy that can provide a functional copy would have a different effect as you're rescuing the cellular function. So based on these different aspects, what is being seen is that generally the sooner the treatment occurs, or where the higher the strength or the functionality of the patient is, you actually get better outcome. And this makes the next point extremely important, which is requirement of diagnosis and screening at earlier time points. So with this, I would like to switch gears to the development of gene therapy programs at Nationwide Children's Hospital. So I work at the Center for Gene Therapy at Nationwide Children's Hospital. Our center was founded by Dr. Jerry Mendel back in 2005, and is being led by our current director, Dr. Flanagan, since 2017. I have had an opportunity and just been honored to be working with my current mentor, Dr. Meyer, since 2017. But this 2017 is when Dr. Meyer joined the center as the assistant professor. I have had pleasure to work with her since 2011 when I was a graduate student with Dr. Kaspar. So our center mainly focuses on developing gene therapies for neuromuscular, neurological, as well as neurodegenerative diseases. So far, we have been involved in 11 gene therapy clinical trials, with 9 of them originated from our own center. Along with that, there is also successful gene therapy startup companies that have been founded from the labs at Nationwide Children's, including Avexis, Myonexis, Milo, and Abiona. So as we work our way through the neurological or neurodegenerative diseases, the central nervous system disorder or the nervous system disorder have an extra challenge of delivering therapies to brain and spinal cord. And that is really because of just the mere size and distribution of the central nervous system as well as the peripheral nervous system. It also has a tremendous diversity of cell types that need to be targeted. And also there is a barrier in order to get to these cell types, which is the blood-brain barrier. So most of the drug therapies, they do not target these cell types very well. However, this was changed with one of the seminal discoveries made by my PhD mentor, Dr. Brian Kaspar, back in 2009, when he showed that the AAV9, which is one of the AAV serotypes, when it is delivered into an adult mouse via tail vein injection, that means the vector is introduced into the blood, it can cross the blood-brain barrier and targets the central nervous system cells, especially neurons and astrocytes in the entire spinal cord and brain. What you are seeing here is the targeting of nervous system cells with an AAV9 that is expressing green fluorescent protein. That's why these cells here are green. With this particular discovery and the subsequent studies in mice, non-human primates, we were able to take this discovery into the clinic and to the patients with a successful translation for an extremely fatal neurodegenerative disease in kids named spinal muscular atrophy, where a single dose gene replacement therapy actually did wonders in the type 1 SMA kids. This study was published in 2017, followed by the approval of AVXS-101 or Zolgensma in 2019. Now, this of course means that AAV9 can be utilized and has a bright future for the gene therapy. However, I wanted to take a moment to actually put the gene therapy in the perspective of doses. So in case of the gene therapy that was actually used in SMA, the intravenous dose that was delivered was anywhere between 6 to 10 to power 13 to 1.2 10 to power 14 viral genomes per kilogram of body. What that means is for a 3 kilogram patient, you actually are introducing 1800 billion viral particles. Now, that definitely is more viral particles than the stars in our Milky Way. And I Googled this earlier to make sure We have around 400 billion stars. So that is why all these kids are the rock stars of everything. But what it actually, the reason why I wanted to actually bring this up is even though the intravenous delivery is possible, it does have its own challenges. And the main challenge being the vector production, because you need so much vector. So you need the facilities that can provide that much vector, the time required for it, and the price that is needed. At the same time, as we discussed earlier, as you increase the doses, which will have to be increased for the kids or the patients that are older, it also significantly increases the safety considerations at high doses, which is why we thought whether we can circumvent this problem by introducing vector in the cerebrospinal fluid. So the cerebrospinal fluid is a fluid that surrounds the central nervous system as well as the peripheral nervous system, and in central nervous system, brain and spinal cord, and it actually flows in and around. It protects the cells that are underneath, but it also provides and transports the nutrients and other components from the central CSF into the cells of the CNS. So what we wanted to determine, can we deliver our AAV9 into this CSF space and whether it actually can have beneficial effects. So we did these studies in multiple disease mouse models where AAV9 can be delivered into a CSF in a day 1 or 2 day old mouse pup. And we were able to see that the delivery of our therapeutic gene in CSF does have beneficial effects in improving the outcome of the disease models. We then wanted to determine, can we translate this into non-human primates? So we performed the dosing and safety studies in non-human primates, in which case we actually target the intrathecal space in the lumbar spinal cord. This is the same technique by you can actually do spinal tap. Just instead of taking the CSF out, you would be delivering the AAV9 vector. So it goes into this CSF space, which actually bathes the entire central nervous system. We also implemented another technique that is known as Trendelenburg tilting, which improves the spread of the vector from the injection site, which is more towards the hip, because that's where your lumbar spinal cord is, or lumbar region is, towards the brain and the upper regions of spinal cord. Now, what you're actually seeing here is again the same AAV9 that has a green fluorescent protein introduced in the intrathecal space in a 1-year-old monkey. What you see here is there is a robust expression of the green fluorescent protein in all levels of spinal cord. We also see that as we actually change the timing of tilting, we do see more improvement in the upper spinal cord regions and even in brain. And one of the main advantage of this technique is these studies were done with 10-fold lower dose as compared to the intravenous study. So that actually gives us what it in a realistic sense means we can now treat 10 patients at the same time as we could have with one intravenous delivery. We also perform these studies in older monkeys as the patients with these diseases actually range from newborns all the way to the adolescent kids. So as you can see on the left side, this is a control brain, different regions of a control animal where there is no GFP immunofluorescence. However, the monkeys that were treated with the AAV9 GFP actually light up really well in different regions of brain as well as spinal cord. So what we have already done, and which is a standard practice when you are developing these gene therapies, is to perform your preclinical studies in the mice, then follow them up with the large animal studies where you can determine the safety of the treatment as well as the dosing, and then move it to the translation into the clinic. With this approach, so far, we have been able to translate our earlier intravenous studies with the GFP to AAV9 SMN, which led to the Zolgensma approval. Since then, we actually were able to switch the route to the intrathecal delivery and have been successful to bring these therapies into the clinic for the phase 1-2 trials for SMA, as well as Batten diseases, and our recent clinical trial that was approved in October of 2021 for SMART1. Now that brings me to the last two topics that I am very fortunate to discuss with you on our efforts for gene therapy for NF2. Now, I am still not 100% sure what to call this disease. So I took the liberty to just call it Neurofibromatosis 2, and I really apologize for that. But what we are looking for over here is the mutations that occur in the tumor suppressor protein Merlin. So our efforts are whether can we utilize the gene therapy approaches to treat the NF2 loss of function cases. So in this case, we are trying to actually bring back a healthy copy of NF2 using AAV9 gene therapy. At the same time, we are also trying a different approach, which involves a phosphorylation resistant copy of NF2. Based on the literature search, even in the normal scenario, a healthy NF2 can be active when it is non-phosphorylated, but after its phosphorylation, it does become inactive, even in the normal scenario. So we wanted to determine, can we circumvent that particular problem if it arises? So in collaboration with Dr. Longsheng Chang at Nationwide Children's and a generous funding efforts from NF2 Biosolutions, we designed 4 different promoters that are human NF2 truncated promoters. So that actually more or less gives us the specificity where NF2 is required, and utilize them with AAV9 vectors. As you can see here, for our studies, we utilized NF2 CDS, or the NF2 coding sequence, which would be our therapeutic vector. But at the same time, in order to determine whether our promoters are working, we are also utilizing green fluorescent protein as the reporter, as we have seen earlier. We performed these studies in vitro as well as in vivo, and we will be, for the sake of today's presentation, we will be looking at the immunofluorescence stainings the same way that we have seen in earlier studies. What we could see after a single intra-ICV injection, where we are targeting the CSF space in a wild type mouse, so these are not the diseased mice, we wanted to determine whether our vector can express the GFP under those 4 NF2 promoters. And we are mainly looking at 4 specific regions of the mouse brain that, based on the literature search and the research already available, are important when it is relevant to NF2. So we look at cortex, hippocampus, basal ganglia, and pons medulla. As you can see, of the 4 promoters, promoter A gives a robust expression in all 4 regions, followed by promoter B, C, and D. These are the magnified images of these regions, where you see the green is the GFP immunofluorescence, and the red is a counter stain for the cells that are in these regions. Again, the promoter A does a great job in a robust and widespread expression in all 4 regions, followed by promoter B, then C, and D. As we are talking about NF2, we also wanted to determine whether this CSF delivery can reach the peripheral nervous system. So we wanted to determine what is the status of GFP expression in sciatic nerves in these mice. And as we could see, both promoter A and B were able to express the GFP, that's our cargo, into the sciatic nerves of these mice as well. Finally, we wanted to determine whether these vectors can express NF2 under these same promoters. And as you can see here, similar to the GFP studies, all 4 promoters do express the NF2 in the different regions of the brain. You do see some staining in the non-injected mice, but that is mainly as the antibodies that we are using do detect some of the mouse expression also. But with completing these studies in the wild type mice, now we are performing these studies in the disease mouse model for NF2, as well as determining the long-term safety and efficacy in the safety studies in the wild type mouse model as well. Now, towards the last part of my talk, I wanted to also introduce the other way we are studying the NF2 disease, which is by developing in vitro models to study NF2. And this is mainly because you all know there is a scarcity of having a reliable cell or animal model, especially for NF2. And we wanted to determine, of course, you can develop more mouse models, but that actually takes a long time. Also, it has to recapitulate the disease phenotype that is happening in the patients. And if you are not able to do that, again, as we mentioned earlier, you're looking at different efficacies in the animal studies versus the patient population. So here we wanted to take advantage of our long-lasting experience in the in vitro disease modeling. So what we are trying to do here is to develop induced Schwann cells from skin fibroblasts that are obtained from the NF2 patients. So in this case, we are taking skin biopsy, which is about 3 millimeter by 3 millimeter skin punch from the NF2 patients or healthy individuals, and directly reprogramming these cells into induced Schwann cells. So far, we have been able to do this with skin biopsies from 3 NF2 patients. And what we wanted to then determine is whether these induced Schwann cells, now, do they have the similar phenotype as it could be in the patients? So the first thing we wanted to determine was the NF2 levels. What we could see is all 3 induced Schwann cell lines established from 3 patients have reduced NF2 protein. This is the Western blot technique where you can specifically determine how much NF2 protein is present in these cells. We also could see that there is a corresponding decrease in the mRNA, which actually makes the NF2 protein in the patient lines. We looked for other markers, especially the Schwann cell markers. And what we noticed is the patient cell lines actually show reduction in the mature Schwann cell markers, which is a hallmark of the cancer cells, that they are undifferentiated. Moreover, we were also able to see that there is increase in some of the stem cell markers, especially SOX2 and c-Met in the patient lines. And finally, we also determined that when we look at the cell proliferation characteristics of these patient lines, 2 out of the 3 patient lines do have higher cell proliferation than the Schwann cells that were derived from the healthy individuals. So with this, it gives us a confidence to utilize these in vitro models for further testing of therapeutic strategies, including small molecules, the antiretroviral drugs, and even gene therapy. Now, one of the challenges that we still have is getting enough patient cells or getting enough cell lines established in order to expand our repertoire for these cell lines, which is where the meeting like this actually is extremely helpful. So in the end, I hope you would agree that the AAV vectors do act as a highly promising tools for gene therapy. It does hold great promise for the treatments of patients with severe debilitating disorders. There are many clinical trials undergoing. However, we are still at early stages. And it has been shown more and more times that the early intervention is the key, which also requires the better and earlier diagnosis. So I would just end my talk with bringing up a few ethical and economic challenges. And these are not to actually show that, oh, this is such a challenge and this will not be overcome. But these are some of the things that need to be kept in mind when we are talking about the gene therapies. The first one being managing expectations. The gene therapies may not cure the disease, but at this point can very well act as a treatment where it actually can slow down the disease or just to stabilize this disease. At the same time, the development of gene therapy all the way from its conception to the market is a very costly affair. To put in perspective, the current price tag for the approved Zolgensma is $2.1 million for a single dose. Now, of course, if you consider the economic burden of the disease itself, which is about $200,000 per year for the entire span of the disease, this is still nothing. However, in the entire context, it is still expensive, which is the same thing when it goes to the drug development cost, the manufacturing cost, which can again take anywhere between 5 to 10 years to develop and can cost about like more than over $50 million all the way from conception, clinical trial to the market approval. And I'm bringing this all up is because we need to have this conversation between the patient families, the patient care providers, the researchers, the companies, the insurance agencies, the legislators in coming together and bring this cost down again for the sole purpose of benefiting the patient life. So with this, I would like to thank all the members of Meyer Laboratory, especially my mentor, Dr. Catherine Meyer, and two of my colleagues, Crizel Alcantara, who is actually spearheading this initiative for the NF2 diseases, and Pipasha Biswas, who is a graduate student at Ohio State University. I'd like to thank all our collaborators, NF2 Biosolutions for the funding opportunity, and of course, our NF2 patient families. And thanks to all of you for being now a extended NF families for having me here and giving me this opportunity to share our progress. Thank you.
Thank you very much for that talk. I don't see any questions in Huva. I'll start out. Oh, Annette. Well, I was just going to ask sort of a question I'm sure is in everybody's minds, but it's not quite your thing. I think everybody's interested in NF1 as well. What can you tell us about where the state of the art is in NF1 gene therapy?
Yeah, so one of the point that was actually mentioned even yesterday when there was a notation for some of the research that Dr. Clapp has been doing for the NF1. One of the main challenge with the NF1 gene therapy is the size of the transgene itself. That transgene being 8 kb, it is difficult to actually put it in the same AAV vector. However, there have been strategies that are underway where you can utilize dual vector system, where you can have a half part of the gene in one vector, the other half in the other vector. Now that comes with its own challenges, that every cell that you actually want to target have to have both, because if you actually have one or other, you are asking for different problems altogether. One of the things I wanted to mention early on is also that when we are talking about the gene therapies, you don't necessarily have to have the exact non-functional gene to be replaced. There are studies that are going on determining if the mechanism of the gene is available, you can try to target the downstream targets that might be still involved, which is one of the ways that Dr. Clapp had tried in the 2019 studies, where it was just a small domain being targeted. But as of now, I think the NF1 gene therapy still needs more development.
So yeah, this is fascinating, very close to my heart. When you do these ICV injections, do you get less liver tox? Do you see less liver accumulation?
Yeah.
Oh, that's fantastic.
Yeah. So one of the things that we have actually seen is AAV9 somehow still reaches everywhere. But what we do see, especially when you compare the non-human primate studies between a intravenous delivery of AAV9 in the monkey and the intrathecal delivery in the monkey, the percent or the, I would say the amount of vector that reaches liver is significantly lower with the intrathecal delivery. It still goes there. So that is why you still need the dose escalation studies and safety studies, but it does reach, but it is lower.
Maybe you could explain for the audience why you asked about the liver and that, just to bring everybody.
Yeah, so one of the big problems with the AAV9 vector, which is kind of your packaging, it's your little bus that brings the gene to the right place. One of the big problems with that is that if you do the IV delivery, which is intravenous, there was a big accumulation of this AAV9 vector in the liver. So for the patients that would get this gene therapy with an AAV9 vector, they would suffer from liver toxicity. And so, in fact, there was a limitation on how much gene therapy you could give because of the bus, not because of the gene. So one other thing that I wanted to mention, and I want to make you aware, is that we're currently funding a non-human primate AAV9 intrathecal, and we should have the response in 3 weeks, I hope.
Yeah.
So we should stay in touch.
Yeah, no, of course.
And then there is one last thing that I wanted to mention is that if you're looking for interesting cell models, we can put you in touch. There is a very interesting group in Plymouth that has a fascinating 3D cell
of
culture for you.
course. Yeah, of course. Thank you.
Marina has it. Dr. Steg.
Thank you. That was a very interesting talk. Thank you. I think, one of the challenges or one of the things we have seen that the gene therapy that so far have been approved are for conditions that don't involve tumors. So with specifically looking at NF2 related schwannomatosis or even NF1, I was hoping you could share with the audience which type of challenges you see for these gene therapies, because, as you said, we are in the infancy. So this is not a treatment that will be available, next year or maybe not even within the next 5 years.
Yeah, I think you brought up a correct, like a very important point when we are talking about the diseases that that mainly involve a certain cell type that you actually know and that you need to target, as compared to some of the other diseases, and especially the NF related diseases, because you actually have such a wide range of tumors that can actually come or that can arise. And that is exactly where we think that having it may not be enough to just have a single modality or a single module intervention. You might actually need multiple. So for our thinking, for example, using a intrathecal delivery for these diseases would be not just to treat the tumors that are existing, but give the cells a chance to have the functional copy before the next tumors arise. So that way you're at least targeting what is already there. But at the same time, you can combine that with intratumoral injections, where now you actually are treating the tumors one at a time, at least the ones that are accessible, and then still preventing what could come next. So that is, I think that's more or less a general idea how we are thinking about these therapies. Of course, it would actually be helpful as we actually move forward with our in vivo studies, because all of these need to be tested in post-animal models, which they still hold the truth for the successful translation.
I think we have time for one more question, if anybody has one. Oh, there, thank you.
Thank you for that really, really great talk. Thank you. I have actually a couple of questions. One is in terms of the immunogenicity. So if the bus is detected by the immune system, is there any thought about using a different kind of bus of a different color that you could use in combination? So you maybe give one bus as the first dose and then use a red bus for the second dose.
Yeah. Okay. Yeah. So you brought up a really good point. So yes, it is possible. However, what is also seen, and that's where the nature is just amazing. These AAVs, as they have evolved, there is still cross-reactivity. So even if you actually, what we have seen in some of these our studies is if you actually take the serum sample for the patients that were treated with AAV9 and you look for the antibodies against AAV9, you will all, of course, find them. But you will also find the antibodies against other serotypes that the patient did not receive.
Yeah.
So the cross-reactivity still is.
So what instead of
Yeah, yeah. So as I mentioned earlier, the antibodies are developed when a vector or a virus enters the system. Our immune system reacts to it as a foreign material, and that's our defense mechanism. So the body will generate these antibodies to get rid of the virus, because whatever we put it inside the virus, for body, it's still a virus. It's still a foreign material. And because of the differences between these different serotypes are so subtle, you can still have the cross-reactivity. So it is possible if you actually have two completely different vectors.
Right.
And that is where this capsid evolution does play important role.
or maybe not use a bus, but use a plane, like
Yeah.
and by that I mean a bacteria, not a virus.
Yeah. I'm not sure if FDA will like it, but
Well, I think there are some studies about bacteria, but also last question, even if you solve the problem of the buses, how long do you actually see the expression?
there are long-term studies with AAV. I know there is a study where they actually had introduced a gene in monkeys under a transducible promoter, and they actually keep on turning it on and off every year to just see if the expression is still there. And I think that study is going on for almost 15 years now. So it is possible to actually see a long-term expression.
I'm going to squeeze in one last question.
For patients who do have antibody development to the AAV vector, has there been any thought about using immune tolerance induction? Like that's from my experience in ERT, where you use large protein replacement therapy for disorders, patients who develop high antibody levels after a period of time to the ERT. What about immune tolerance therapy using rituximab and other drugs that, have been really successful? So what about that principle for this?
Yeah, so one of the things that has always been challenging is all these therapies they are pretty severe when it comes to actually administering them. And that is where, because we don't really know how much the benefit of that is going to be, because along with the antibodies, there is still a adaptive immune component where you're talking about the plasma cells or the memory cells that you cannot get rid of directly. So there it's still a little bit of reservation. Yes, it is actually in discussions, but I don't think we are there yet. And so I'll just make one last point as we are mentioning about the liver toxicity. There have been studies that where you do see a more liver toxicity. As far as the Zolgensma study was actually concerned, those kids, they actually received a prolonged immunosuppression treatment also with prednisolone. And during that duration of that study, we did not see any liver toxicity all throughout. So just wanted to let you know that there are clinical trials where it is still a concern, and that is why it is still being monitored all throughout for the serum chemistry and the blood chemistry for these levels. But so far, at least the studies that we have performed at Nationwide Children's, we have not seen occult liver toxicity in any of our gene therapy studies. Thank you so much.